Imagine this room a cave. Probably the first thing that comes to mind is darkness. But for me, what I encountered was the chaos of silence. Not because nothing's there, but because every sound feels close. From water dripping from the rock, the flutter of bat wings overhead, the sound of my own breath. The air is warm and heavy with humidity, pressing in on you until the space itself feels tight. The ground beneath your feet can be dry in some places. In others, it can be slick, with water dripping from the ceiling and accumulating in low places. And in these caves, there are pools of water where you can see them. Tiny translucent shrimp that flicker in and out of view in water that has never known sunlight. But more on those later. In Mexico's Yucatán Peninsula, you can experience this very same system in a different way. Standing at the edge of a cenote in the middle of a jungle. A cenote is a sinkhole which forms when limestone collapses and exposes the groundwater below. In the Yucatan, there are no surface rivers or lakes. Instead, groundwater moves through limestone, moving through cracks and crevices, linking thousands of cenotes across the peninsula. So most people will experience these systems from the edges, beside a cenote or entering a dry chamber of a cave. But there is a third way to experience it, by entering the cave passages themselves. Cave divers have mapped and explored portions of these systems, and through those explorations, they have found cathedral-like chambers, winding limestone corridors, and animals that are found nowhere else on Earth. But despite decades of exploration, only a small fraction of this aquifer has been mapped, leaving most of it beyond human reach. As you move to the coastlines, things become even more complex. Seawater pushes in and meets fresh water that is flowing from the interior. In this video, as you watch the diver, you can see a shimmer. That shimmer is the mixing of fresh water with seawater. Similar to when you mix oil and vinegar, the densities of those two bodies naturally remain separate. So 1 minute, you can be in fully fresh water and then abruptly shift to seawater. In these systems, there is no sunlight. The water is crystal clear, indicating that there are very low food resources. In some areas, they are very low in oxygen, making this a challenging place to live. But despite all that, life thrives here. So what can life in this hidden world teach us about connection, resilience, and adaptation? Let me introduce you to Tifladia, a group of translucent shrimp that are found in groundwater systems in several parts around the world. They're only about a few centimeters in size, and no, you cannot eat them. I get that question all the time. But in the Yucatán Peninsula, something remarkable has happened. Here, 5 distinct species have evolved, and to the untrained eye, they look nearly identical, but genetically, they are profoundly different, and that difference tells a story. 4 of the 5 species are specifically adapted to freshwater. They occur throughout the aquifer, throughout the peninsula, and using genetic data, we have found that populations are connected from inland regions to coastal caves. But wherever they're found, they're confined to that freshwater layer. So their physiology and evolutionary history is shaped by being confined to narrow chemical conditions. The 5th species was different from the beginning. Tifladia zilamensis was first described as a saltwater cave shrimp. But when we started to analyze its DNA, the plot thickened. I remember the first time that I was looking at the genetics results, and I was looking at a map dotted with sampling sites. And one thing you notice is that this species is always found along the coastlines. And also, their populations were connected over vast distances as well. But one thing that confused us was that some of our samples, at least based on the DNA, it was telling us that some of the samples were collected from freshwater in addition to the saltwater. So, at first we thought perhaps did we contaminate our samples? Because salinity establishes a very hard boundary that most species cannot cross. Freshwater species stay in freshwater, saltwater species stay in saltwater. But what the data showed was that zillamensis is not confined to that saltwater layer, but instead it crosses that hard boundary and moves into freshwater. So to put this into perspective, near the Tudor's town of Tulum, we've collected samples from 4 cenotes. So this diagram shows basically a cross-section showing you the connected cave system. And these 4 cenotes are found within a 5-mile stretch, starting from near the coast and moving inland. And here, remarkably, we find all 5 species, something that we didn't see in any of our other sampling sites. But here, we always found those first 4 species in freshwater, but here is where we saw zillamensis, not only in the saltwater, but crossing that barrier and into freshwater. 5 species, 1 aquifer, overlapping ranges in some areas. The data was showing us that populations were connected over vast distances. Not only that, it was also showing us that the different water layers that are thought to be isolated are also connected biologically. But adjusting to environmental changes was only part of the story. In that saltwater layer, there is another inhabitant, a remipede called Shibalbanus tulumensis. This is the only known venomous crustacean on Earth. And in this saltwater layer, it actively hunts zillamensis. And here, despite all this, zillamensis persists. So not only does zillamensis cross this salinity boundary, as it's moving from one layer to the next, it has to adjust to different densities, differences in oxygen content, food availability, all this while navigating active predation. In this aquifer, we see 3 different evolutionary strategies. We see some species confined to narrow chemical conditions. We saw this in the freshwater species. We also see this in the remipede in that saltwater layer. But the remipede is a top predator in that saltwater layer. And zillamensis navigates between those 2 worlds, trying to avoid predation while also adapting to environmental extremes. So what appears isolated on the surface is deeply connected below. And those connections have consequences. Cenotes are a source of fresh water for millions of people across the peninsula. It has sustained the jungle, wildlife, and communities that depend on them every day. But as the region continues to develop, what happens on the surface doesn't stay there. On this map, it shows major cities across the peninsula. Today, they are interconnected by a major railway system, the Maya train. And as we overlay our sampling sites of Tifladia, you can see there is quite a bit of overlap in the distribution of the shrimp along with that train system. And why am I mentioning this? It's one thing to build a road where you're paving over the limestone. But to sustain a railway system, you have to drill a hole straight into the limestone and insert a concrete pillar that enters a space like this one and into the groundwater itself. So as the region continues to expand and urbanize, we continue to see increased pollution, pesticides, sewage, industrial runoff, all entering the system. And once it enters, it has nowhere else to go. It will stay there. But groundwater is not stagnant. It gradually still moves through that limestone, eventually bringing those contaminants far away from where the contamination first began. And remember, I shared with you that there are species that are confined to narrow chemical conditions. They may not be able to adjust when big changes like this come their way. And even though we have species like zillamensis that appear to be more flexible, that are able to cross a boundary and maybe seem to be tolerant to change, but tolerance does not equal immunity. Even a flexible species has its limits. This aquifer is also shaped by deep human history. The cenotes were not just sources of water. They were also, especially for the Maya people, they were also sacred spaces, places of ritual, burial. And now it preserves some of the most important historical records in the region, from human remains, fossils, artifacts, and ceremonial objects. So studying DNA that moves through passages, it gave us a window into connection, into connectivity, indicating that both organisms and water share the same system that has sustained both ecosystems and human societies for generations. It was also teaching us that perhaps preserving one cenote can help preserve them all, because cenotes are not isolated features, they're all part of one living, interconnected aquifer that quietly links nature, culture, and future generations. The more I studied Tifladia, especially zillamensis, the more I realized this wasn't just a scientific discovery, it was a lesson. Tifladia zillamensis is found in parts of the aquifer where it's anything but stable, where fresh water and seawater meets and predators patrol the dark. This same aquifer is also shaped by human pressures. What happens on the surface can enter this system below. Resilience here isn't just about stability. It's about adapting in the face of chaos. And human societies are not so different. Communities, relationships, institutions, they're all layered and interconnected. And when one part of the system is harmed, that harm is not contained. Instead, it trickles outward, moving out like the shrimp or even contaminants move through the groundwater. Some people stay within their comfort zones. Others push through their comfort zones in the face of uncertainty. Connection, not isolation, is what determines whether systems endure. But adaptation determines whether we survive it. Studying Tifladia, or studying these shrimp, has shown us that resilience doesn't have to be dramatic. Sometimes it could be small, quiet, even invisible. It has also shown us that even in chaos, life in this hidden underworld finds a way to adapt, connect, and endure. And if a tiny shrimp living in complete darkness can navigate a changing and uncertain world, then perhaps so can we. Thank you.